Hello everyone, welcome back to Prebiotics Nutrition. Today we are diving deep into specific prebiotics. We'll explore three key types: inulin, fructooligosaccharides or FOS, and galactooligosaccharides or GOS. We shift our focus and dive deeply into the first of our three key prebiotics, inulin. Inulin is a fructose-based polysaccharide found in various plants such as chicory roots and is recognized for its potential in both industrial applications and health benefits. It serves as a renewable source of fructose for producing biopolymers like polyhydroxybutyrate or PHB, and plays a significant role in modulating gut health and disease prevention. The following sections explore inulin's applications and health impacts based on recent research findings. Inulin can be hydrolyzed and fermented to produce PHB, a biodegradable polymer. The process involves using inulinase to break down inulin, followed by fermentation with Cupriavidis necator, resulting in a polymer with improved thermal stability compared to those produced from pure fructose. Inulin consumption significantly alters gut microbiota composition, promoting beneficial bacteria such as Bifidobacterium. This change enhances gut health by increasing short-chain fatty acids or SCFAs, which are crucial for maintaining gut epithelial integrity and immune function. Inulin's impact on the gut microbiota also extends to reducing obesity-related disorders. It decreases body weight and fat accumulation in high-fat diet-induced obese mice, with these effects being partially transferable through fecal microbiota transplantation. Inulin supplementation has been shown to inhibit the growth of estrogen receptor-negative mammary tumors in mice. This effect is linked to changes in gut microbiota and epigenetic regulation, suggesting a potential strategy for breast cancer prevention. Maternal inulin intake can alter the offspring's gut microbiome, potentially reducing asthma symptoms by increasing short-chain fatty acid-producing bacteria and modulating immune responses. While inulin demonstrates promising benefits in various domains, its effects can vary based on individual microbiota composition and dietary habits. Further research is needed to fully understand its mechanisms and optimize its use in both industrial and health-related applications. Dietary supplementation of inulin contributes to the prevention of estrogen receptor-negative mammary cancer by alteration of gut microbial communities and epigenetic regulations. Breast cancer is a common malignancy in women in the United States. Inulin, a health supplement, may influence breast cancer onset and progression. This study investigates inulin's effect on estrogen receptor-negative mammary carcinoma in mice. Inulin supplementation significantly inhibited tumor growth and delayed tumor latency. Mice on inulin had a distinct and diverse gut microbiome. Plasma propionic acid levels were higher in the inulin group. Expression of epigenetic modulating proteins decreased with inulin administration. Sodium propionate also showed breast cancer prevention effects through epigenetic regulation. Findings suggest inulin may be a promising strategy for breast cancer prevention. Breast cancer is frequently diagnosed in women globally, with rising incidence and mortality. Risk factors include age, genetic mutations, and environmental factors. 20% of breast cancer cases are estrogen receptor negative, highlighting the importance of prevention. Healthy diets rich in fiber can reduce breast cancer risk by affecting gut microbiota. Dietary fiber is linked to lower circulating estrogen levels. Gut microbiota regulates homeostasis and may influence breast cancer risk. Inulin, a prebiotic fiber, promotes beneficial gut bacteria and short-chain fatty acid production. Prebiotics like inulin may mediate cancer prevention through inflammation and epigenetic changes. Dietary interventions can reverse epigenetic modifications linked to tumorigenesis. The HER2/νu transgenic mouse model was used for studying estrogen receptor-negative tumors. Inulin concentrations of 8%, 10%, and 15% were tested for cancer prevention. All inulin diets reduced tumor incidence compared to control. 8% and 15% inulin significantly decreased tumor volume and weight. 15% inulin notably delayed tumor latency and onset. Inulin promotes health-benefiting bacteria growth. Gut microbial composition was analyzed. Fecal samples were collected before and after tumor onset for analysis. Higher microbial diversity was observed in the 15% inulin group before tumor onset. Significant changes in microbial composition were noted between treatment and control groups. Alpha diversity metrics indicated higher diversity in the 15% inulin group. Bray-Curtis beta diversity analysis showed distinct clustering of samples. Relative abundance of bacteroidates decreased while firmicutes increased in treatment groups. Inulin diets increased microbial diversity compared to control before tumor onset. Tumor development altered gut microbiota and overall metabolism. Alpha diversity in inulin-treated groups was not significantly different from control post-tumor. The firmicutes to bacteroidetes ratio decreased in inulin-treated groups after tumor onset. Tumor development reversed some inulin-induced microbial composition changes. Short-chain fatty acids profiles were measured to assess the impact of inulin on tumor growth. Propionic acid levels significantly increased in the 15% inulin group. Other short-chain fatty acids showed varying levels but were not significantly different. Inulin treatment affected protein expression of epigenetic modulatory enzymes. Significant reductions in HDAC8 and DNMT3B expression were observed. Inulin diets inhibited key epigenetic regulators in mammary tumors. Propionic acid increased in the 15% inulin group, was tested on human breast cancer cell lines. Sodium propionate exhibited dose and time-dependent inhibitory effects on cancer cells. Non-cancerous cells showed little to no toxicity from sodium propionate treatment. Sodium propionate significantly decreased tumor incidence in C3, tag mice. Treatment delayed tumor development and reduced tumor weight, though not significantly. Sodium propionate treatment reduced expression of HDACs and DNMTs in tumors. Increased expression of tumor suppressors like p53 was noted with treatment. Current breast cancer treatments are limited for estrogen receptor-negative patients. Dietary interventions are promising. Inulin supplementation showed significant anti-cancer effects in mouse models. Changes in gut microbiota composition were linked to inulin treatment. Tumor development negatively impacted beneficial gut microbiota changes induced by inulin. Short-chain fatty acids produced by gut bacteria may mediate protective epigenetic changes. Inulin's potential as an HDAC inhibitor suggests new avenues for breast cancer prevention. The study utilized HER2/NU and C3, tag transgenic mouse models for breast cancer research. Mice were maintained under controlled conditions, and protocols were approved by relevant committees. Inulin-supplemented diets were prepared with varying concentrations for testing. Sodium propionate was added to drinking water for evaluating its effects on tumor development. Tumor metrics were recorded weekly, and samples were collected for analysis. Plasma and tumor samples were prepared for further biochemical assessments. Fecal samples were analyzed for microbial composition using 16S rRNA sequencing. Diversity indices and dissimilarity measures were calculated to assess treatment effects. Plasma short-chain fatty acids were quantified to evaluate the metabolic impact of dietary treatments. Standard operating procedures were followed for accurate measurement and analysis. Protein extracts from tumors were analyzed for expression of key regulatory proteins. Antibodies were used to probe for specific proteins involved in tumorigenesis. Various human breast cancer cell lines were cultured and treated with sodium propionate. Non-cancerous cell lines served as controls for assessing treatment effects. While inulin demonstrates promising benefits in various domains, its effects can vary based on individual microbiota composition and dietary habits. Further research is needed to fully understand its mechanisms and optimize its use in both industrial and health-related applications. Please subscribe to our channel for more informative videos on health and wellness. Don't forget to hit the bell icon to get notified about our latest uploads. Thank you for watching, and we hope to see you in our next video.